Welcome, everyone, to the 10th Annual 2021 Student Research Symposium. My name is David Cox, and I'm the Director of Skene Library, OSL, and the SRS program. The SRS program is an annual campus-wide event that provides both undergraduates and graduate students the opportunity to present their research in a variety of formats: poster sessions, oral presentations, and 3-minute speech competition. We hope you'll join us for these events over the next few days, and we thank you for joining us today for our second event, which is Poster Session 2. The SRS is grateful to Tech faculty member Dr. Joel Charbrough and also Dr. Stephanie Plant, who are helping with us this as moderators or coordinators, and we thank them for their assistance today. I know that Dr. Charbrough is going to be moderating today's session, so there will be some directions for him and for you in just a moment, okay? And we also thank the help of SRS staff members who are assisting with the tech, and we believe everything will run smoothly. Now, during normal times, the SRS would take place in person on our beautiful New Mexico Tech campus. But because of COVID-19, we're here in the virtual world, and we did virtual last year in 2020 and again here in 2021. This session is a poster session. Several New Mexico Tech students will have the opportunity to present their research to you, and you will be able to ask questions. In a moment, Dr. Charbrough will start things off, but first, a brief word from one of our SRS staff members.
Thank you, Dr. Cox. So hello, everyone. My name is Katie Teams Norris. I am an SRS staff member and a staff member at the Office for Student Learning. So thank you for being here today. And because we have a lot to cover, this is how today's session will unfold. So each student or group will give a brief presentation based on their poster, and then the moderator will facilitate questions from the audience. So during those presentations, all of you will be muted, so that way there's no background noise or any unexpected interruptions. But when the time comes for the Q&A, question and answer, you will be able to unmute yourself and ask questions freely. And of course, the chat box will remain open throughout the entire session, so you can put any questions you have there as well. And if you need any technical assistance during the course of this event, please feel free to send a direct message to myself or Sophie. And when Dr. Stephanie Plant arrives, you can also feel free to send a DM to Stephanie as well. So just for ease of use and access, the moderators, volunteers, and presenters are going to be at the very top of the participant list. So just a couple of things before we begin. We very much hope that there are no technical difficulties or any problems with internet access today. However, if we do experience any problems, just please be patient as we try to work through it. So if there's a power outage or any sort of unexpected event, we will contact you via email apart from the meeting if you have already provided one to us. So here's one suggestion for the session itself. You might want to change your settings to speaker view, which makes the visuals and presenters the main thing on your screen. And from time to time, we'll be posting the link to the poster gallery in the chat box so that you can take a look at the posters throughout the session. And finally, at the end of the session, we are going to share a link in the chat box that allows you to evaluate today's presenters. So we hope that you fill out that evaluation form as often as you are comfortable, and you'll need to fill out a separate form for each poster and group. So thank you again for joining us, and let's begin. Dr. Charbrough?
Thank you, and thank you to all the SRS staff who have helped organize and are running the tech. It takes a lot off our burden to run this. So thank you all for joining us today for the second poster session. We have 12 posters being presented by 28 presenters during this session. And please keep an eye on the chat where there will be a link to each poster. We're also going to share the screen of each poster while the presenters are presenting. And luckily, the presenters are here with us live, and they'll be here to answer questions and everything. So how this is going to work essentially is that each poster will get time to present their poster, ideally something on the order of 5 minutes or so, and then we can answer questions. At about 8 minutes, it's about time to wrap up so that we can move on to additional posters. So I will, at that time, I'll make sure that we can move it along either via questions or not. Hopefully at the end, if there's any unanswered questions, there will be time to answer those as well. So without further ado, let's get started with our first poster and presenter. This poster is fabricating a remote controlled combat robot and creating a display for a BattleBot arena, presented by Mason Kasperowitz. Go ahead, Mason, take it away.
All right, sounds good. Is my audio coming in well?
Works well, thanks.
All right. So to provide a brief overview, over the course of the semester, I was able to work on 2 projects in the general realm of combat robotics. The first of which was the development of a standardized, simple RC combat robot design, which would be used in a recruitment seminar at UNM Valencia, I believe, and potentially high schools in the future. So for this project, the objective was to design a simple combat robot which could be built in under an hour by STEM-oriented high school students. It was supposed to convey basic circuitry, wiring, programming principles while still being reasonably complex. So to ensure it could be built in under an hour, we used a simple differential steering setup whereby 2 DC motors were controlled via a LN298 motor driver, and the signals for the motor driver were provided by a standard 2.4 gigahertz RC controller. During initial testing, however, it was realized that the remote controlled setup could not be used initially as intended because it is configured for throttle and steering as opposed to 2 separate motor drives. So that required the use of an Arduino conversion system. So an Arduino Nano was connected to the radio receiver, and it needed to decode the control signals back into standard radio signals. And then these standard radio signals can be converted back into new PWM signals to control our design. So the actual coding made use of interrupt service routines to detect the changes in the PWM pins. And then several series of logic and algebra were used to convert these decoded PWM signals into differential steering signals. So in the end, the robot did prove successful. However, there are several potential augmentations that could be added in the future. One of which is there is a third unused signal on the radio receiver, which could be used to control a weapon, such as a spinning blade. And the robot could also be equipped with a reverse function. And it does not have one currently because there was initially some confusion over negative PWM signals, but over the course of the project, which was 2 weeks, we were eventually able to find a workaround. So the second project was my main project for the semester, and that was the programming and wiring of a digital display for the recently completed NMT Combat Arena. So per the SPA RC guidelines, which NMT now follows for its combat robot competitions, there are several sort of timers, stopwatches, and warnings that have to be conveyed to the audience. So it was theorized that a large LED array could be used to display these countdowns, as well as some LED bulbs to display the warning lights. So the system had to be portable, i.e. it had to be able to be disconnected from the arena when the arena had to be moved from place to place. And it also had to be independent of a computer. So the Arduino that would be controlling the LEDs also had to read a series of push button based inputs. So for it to remain portable, it was designed around a signal transmission system. Initially, it was going to be Bluetooth, but that proved difficult. So it was decided to use direct wiring, whereby the control box was connected to the Arduino with a DB9 cable. So initial testing did prove that the system was working as intended. The digits were controlled via special custom drivers, and this allowed a simple serial signal to be converted into the 8 logic signals needed for each of the separate digits. And it was able to be used during the NMT competition on April 3rd of this year, and it provided the necessary information. So in the future, however, it could be augmented by reading an external clock, because it is currently programmed using delays, which was simpler in terms of logic, but it would be more thorough if it were programmed using an external clock. So that is the overview.
All right, thank you so much, Mason. Is there anybody who wants to ask a question? If so, please go ahead and unmute yourself and ask Mason about his project. I have a question, Mason, if you'll permit a very simple question from
Sure.
a person who doesn't understand any of this wiring. But you said that the Bluetooth was difficult to get to work. I mean, Bluetooth seems like magic to me. So
Yes.
why was it difficult? I mean, how is it even like, how were you envisioning it, and why was it too difficult to manage that?
So originally, because the displays would have to be like 12 feet apart from where the judges would be, we needed a signal to get from the judges to the displays. And Bluetooth was originally thought to be the best because we wouldn't have to worry about resistance drop over 12-foot wires, if that was going to be an issue. But it was too difficult because we would need a single Bluetooth module to respond to 2 receivers, or to send signals to 2 receivers. And to do that requires messing with the firmware of the devices, and there's no actual interface with these Bluetooth modules. You have to program them especially through the Arduino serial monitor, which is complex. So,
I see.
Yeah, it was going to be much more reliable to use direct wiring.
Right. Yeah.
Yeah.
It would be cool, but I can see why you made that decision, and I think that makes a lot of sense. Cool. Thank you.
Yep.
Any other questions?
I'm wondering what you have to do to go from this stage to actually getting it into your combat robot, and how that wires up, how that the signals work and everything. I imagine you're going to create a robot to have this work in.
To create a
Combat robot?
yes. So this the display was an augmentation to the
Right.
NMT Combat Arena. So there were 2 senior design teams that competed this year, and the robot that I designed is sort of a recruitment tool, and it's eventually hoped that high school students will be able to compete in the arena that was recently designed. So if that's the question you're asking.
That helps me a great deal. Thank you.
Sure. I saw, I think, one in chat. What's the next step to manufacture and distribute these? Currently, there is an assembly line that will be starting on Monday. We have to solder 400 motor wires, upload 100 programs to the Arduino, and package up all the kits. It's supposed to be as quick as it can be while still conveying information and having something fun and successful that's STEM related for students to do. So, yes.
Cool. Thank you, Mason. Also, Joaquin also said, I like the NMT logo BattleBot. I agree. Super cool.
Yeah.
All right, let's thank Mason. Well done.
Thank you.
And move on to our next presenter. Presenters, Ian Hayhurst, Juliana Keniser, Jordan Zayas, Diego Chavez, and Mateo Gabaldon. They'll be presenting a poster, wireless communication of millimeter wave sensors. Go ahead and take it away. Ian, go ahead. We cannot hear you. Can you
Okay,
unmute?
I just
There we go.
got host permission to unmute, so I was.
Now we can hear you.
Alrighty, nice. Okay, so like you said, this is the wireless communication of millimeter wave sensors. So we were given a project by Omnius. The original purpose of our project was to sort of design a predictive algorithm, but that ended up sort of falling through. And so we decided to find a way to make these sensors communicate wirelessly so that we can have a network of them set up in the house. Now, what this device, the Care Companion, does is it tracks your movements using a point cloud, and it's meant for like elderly or disabled people, and it detects whether or not they've fallen down or hurt themselves in their home, and can send notifications to family members, emergency services, or other resources depending on the severity of what's happened. But what we focused on was getting these sensors to actually be able to talk to each other. So the current setup is just a millimeter wave sensor from Texas Instruments attached to a single board computer. And what this does is it kind of reads the room using millimeter waves, and then it can detect distance, objects in the room, and people, and it can tell whether or not you're standing up or down. But yeah, Juliana, why don't you give us background on this?
All right. So the elderly population is one of the fastest growing populations in the world, thanks to healthcare and modern medicine. Oh, it looks like the poster.
Can we go back a slide? We didn't move forward.
You went back
Thank you.
slide.
Go ahead, Juliana. Sorry about that.
It's still on the BattleBot poster.
It's back on the
Thank you.
normal one for me.
Oh, it's back on mine now. My internet is real slow. Gotta love Socorro.
Indeed.
And so it's very likely that this elderly population is only going to grow. And so falls, as you get over the age of 65, can be very costly. They can cost thousands of dollars and severely decrease your quality of life. And so the point of this is to make this system that Omnius is trying to distribute more affordable. So if it's wireless, then you won't have to install wires and cables in the home potentially, which is very invasive, which is something that a lot of people don't like. Surprise, surprise. It also reduces the cost for having the system, because if you make it more affordable, more people are going to be able to use it. It also, because of the lack of wires, if someone wants to just try it out, they're not going to have the potential hazard of tripping over the system, which seems like a bit of an oxymoron. So it has a lot of positive effects on the system, and if the system is widely adopted, it could have a lot of positive effects on the quality of life for a lot of the elderly and disabled populations.
All right, Jordan, would you like to take the experimental section?
So as far as what we were doing for our experiment, we're basically trying to do each piece of it sort of one by one. And so we have we're using the development boards that Ian mentioned earlier. These have Bluetooth capabilities, of course. And so far we have basically gotten every part of the system working except the sensor to development board. So we have wireless communication between the development boards. We also have communication from the development board to a PC. And it's the last step that we are still trying to figure out.
All right, Diego, do you want to take results?
Yeah, so we ended up getting the 2 microcontrollers that were connected to the millimeter wave sensors working for one-to-one communication wirelessly. And the way we did that is we set up PuTTY. It's another serial peripheral program on your computer that's free. And we used that and we downloaded that and we connected the 2 microcontrollers together to see if they would send packet data back and forth. And as you can see in the lower picture right there, the left-hand side is the peripheral microcontroller, and the right-hand side is the central node microcontroller. And there's buttons on these CC26 microcontrollers that have options. And what we did is we scrolled through those for options to send different hex values to see if it would send data packets. And eventually we did. And so we can send this information over UART to the single board computer that we have for the Care Companion. And this shows that we can have this wireless communication between the microcontrollers that are set up to the millimeter wave sensors.
All right, Mateo, would you like to take the discussion?
So with the microcontrollers establishing Bluetooth and connecting to each other, we have shown that wireless communication can be achieved via the microcontrollers and the millimeter wave sensors. With this, it shows that it won't be as invasive to the actual what's it called? I'm sorry about that. The patient's homes as it is, and with the communication being wireless, everything can be turned off, even every sensor. So if a person decides to go out of the room, everything can be turned off. And with this, the PuTTY also shows that data is being sent. And with this, it overall reduces sensor-to-sensor communication through a hardwire setup, which in turn can create less wires and less trouble with communication and reduce cost.
Yeah. And so for future work on this, the first plan of action would be to finish the sensor to microcontroller communication so that we can actually transmit the serial data that the sensor is collecting. And after that, we would work on, it's a millimeter wave sensor, Joaquin. And after that, I lost my train of thought, apologies. Oh, after that, we would create a star network, which is something that's unique to TI boards. And basically it allows for an easier time communicating between 3 or more different microcontrollers. So this would allow us to set up sensors in separate rooms so that each room would be able to be covered by this millimeter wave sensor so that there's not an unsafe place in your house. And is it possible for unit sensors to be available for seniors who may? Yeah, I mean, you could do outdoor units as well. So the way the current Care Companion is set up, it's actually in a contained, like 3D printed box. So it could be fairly protected from the elements as long as you could run power to it outside. So as far as the AI algorithms go, that's mostly on Omnius's end, because originally our project was a little bit more computer science heavy rather than electrical engineering. Yeah, it is feasible to detect outside though. As far as more future work goes, I think the last thing we would do is probably create a simple system to kick the sensor on when you walk into the room so that it's not constantly transmitting data that doesn't need to be transmitted. But other than that, those are the few things that we would work on in the future for this project. Would anyone like to share any questions verbally?
All right. Thanks, Ian, Juliana, Jordan, Diego, and Mateo. I'm sorry to cut you off. We have to move on for
no problem. Sorry
the
about
sake
that.
of time. But if there are additional questions, you can answer them in the chat, perhaps, or
Yeah,
we'll
no
have
problem.
time at the end as well for some open source Q&A. Thank you guys for a great presentation. All right, and our next presenter is Angelica Cave, and she's going to be presenting about antibiotic resistance genes and bacterial community analysis in the Socorro, New Mexico wastewater treatment plant and surrounding environment. All right, Angie, go ahead and take it away.
All right, thank you. All right, so not to restate the title, but we were looking at antibiotic resistance genes in the Socorro wastewater treatment plant and analyzing the bacterial community to see how wastewater treatment might affect the bacterial community, going from raw sewage to treated wastewater. So to begin with, antibiotic resistance is kind of a huge global health issue. The CDC most recently stated that it's expected to cause more than 2.8 million infections, billions in lost productivity and healthcare costs, but most importantly, over 35,000 deaths per year in the US alone. And one of the biggest problems in antibiotic resistance is carbapenem resistance among the gram-negative rod-shaped bacteria. These bacteria are like the bacteria that are in your gut mostly, so they would be present a lot in sewage, let's just say. And carbapenems are penicillin-like antibiotics, meaning that they have a ring, an organic ring that gives them their efficacy, it's their pharmacophore. So the way that these bacteria are immune to carbapenems is through the production of beta-lactamases, which break the what's called the beta-lactam ring, and it completely destroys the efficacy of the antibiotic. It's completely just inert at that point. This is a huge problem because carbapenems are last line of defense antibiotics against multi-drug resistant gram-negative infections. So once the bacteria are resistant to carbapenems, they're resistant to pretty much everything. The reason that we wanted to look at a wastewater treatment plant is that it's one of the most obvious conduits from the human community into the environment. So things like hospital, effluent wastewater carrying perhaps very drug-resistant bacteria all come together, especially in small, in wastewater treatment plants serving small communities. And the bacteria are able to do what's called horizontal gene transfer between each other. They can do this between very distantly related bacteria, and this allows them to spread antibiotic resistance throughout the whole community. It's really hard to see, but in figure 1, this is a map of the, or several maps of the 5 most common or most important carbapenemase genes, which are listed there. We're not going to go over each one, but I do just want to let you know that the blah in front stands for beta-lactamase, and then the subscript is the name of the, or the abbreviation for the enzyme. The big 5, as they're called, are actually tracked in the clinic, and that's why we have data for them. This map comes from the New Mexico Department of Health. We wanted to also look at any other potential carbapenemase genes that aren't tracked in the clinic, and so we added the gene for the enzyme JES. For the methods, what I did was sample over the course of 9 months from July 2018 to March 2019. And if you look at figure 2, again, hard to see, but each of those little dots is one of the sampling points. The upper image is of the wastewater treatment plant itself. And the N is the influent, AD stands for aerobic digester, which is a step in the treatment process. And EF stands for effluent, and that's a treated wastewater. I also looked at the kind of waterway that the wastewater treatment plant expels the treated wastewater into. So I took samples upstream from the wastewater treatment plant, so a sample that would have no possible effect from the plant, and then a sample downstream from the wastewater treatment plant, assuming that that would show any effect that the wastewater treatment plant would have. I extracted the DNA from communal samples by first vacuum filtering each one to kind of condense all the cells into one place, and then extracted genomic DNA from the whole sample, and then did classical PCR for each of the 6 genes. We also use this genomic DNA for 16S, our DNA sequencing, to determine a kind of broad taxonomic picture of the bacterial community. So in the results, the color coded, so hopefully it makes sense. This shows where each of the genes were detected and the dates that they were detected on. You can see, most importantly, that the most genes were detected in the effluent, interestingly enough. Upstream from the wastewater treatment plant, 2 genes were detected. And if you look at the first gene from each one, I don't know if you can read them, they are very small, but BLAGS is actually present on most sampling dates in all of the samples. So that was very surprising. We also looked at like I said, the community composition. And what I really want you guys to focus on is this little green piece of the pie on the left-hand side. That's the influent. And the green piece are the bacteria called proteobacteria. This is the phylum that the hosts of the genes that we're looking at typically reside in. And to bring it in closer, to look at the classes, the gamma proteobacteria, or that bright green piece of the next pie chart over, is the gamma proteobacteria, which is a class that these bacteria reside in. So between the influent and effluent, you can see a marked increase between the in the proteobacteria and in the gamma proteobacteria. So wastewater treatment likely kind of, if not selects for, then like enriches the type of bacteria that are carrying these genes. And since the most genes were found in the effluent, we know that they can survive wastewater treatment. So with that, I'd like to say thank you guys for listening and see if anybody has any questions.
Wonderful, thank you, Angie. All right, I'll open it up to questions. I have a question too, but I'll limit myself in case somebody else wants to ask it first.
Okay.
I have a question.
Yeah.
Go for it, Joey.
So you're able to detect these genes, and we understand that there's bacteria that are present in the environment that are potential hazards. So are there like means to mitigate this spread, or to prevent like someone like me from getting like sick?
Basically, from what I've found, the ways that you can treat wastewater to actually remove these genes and not select for those bacteria in particular are extremely expensive. So likely removing it at the source, so like at a hospital or at a long-term care facility, would be the most effective, but also probably not very cost efficient. One way that you can stop people from getting sick but not spreading the antibiotic resistance into the environment is to survey environments and see what's there. That's not done in very many places, and in recreational waterways it really should be, especially in New Mexico where we have limited water resources. So it's kind of dicey where all the water comes from in like the lakes that we fish in and stuff.
Thank you, Angie. We have time for one quick question if there is one. Otherwise, I'll put my question into the chat, Angie, because I don't think it's quick.
Okay.
Any other?
Great presentation.
All right. Yeah, agreed. Thank you, Nathan. I agree with you. And Angie, thanks a lot. Really appreciate it. And go ahead and move on to our next presenter, which is Cassandra Velarde. And she'll be talking about exploring surface water as the reservoir of CRE-infecting patients in Southeast New Mexico.
Okay. Is everything good on your end?
You sound great. Go ahead and take it away, Cassandra.
Okay, sounds good. So yeah, not to reiterate the title again, but I'll be talking about the research I've been doing over the last couple of months for exploring surface water as the main source of the dissemination of antibiotic resistant genes, specifically CRE genes. Angie's work kind of relates to this, so please bear with me. There's going to be a lot of terms that are used and that are the same, and it sounds pretty similar, but they are different, I promise you. So to give a little bit of background, carbapenem resistant enterobacteriaceae, or CRE, is an emerging group of antibiotic resistant pathogens. These pathogens include E. coli, Enterobacter, and Klebsiella, and they produce these enzymes called carbapenemases that hydrolyze the beta-lactam ring of the carbapenem, or last resort antibiotic, making this drug ineffective. The bacteria can cause infections, including pneumonia, bloodstream infections, UTIs, wound infections, and all these things that you don't want, right? In 2017, it was reported through the CDC that there were about 13,100 hospitalizations and 1,100 deaths in the United States alone. So globally, this is a concern. On this project, I'll be focusing more on a possible secondary source, which is the environment, because this usually happens in like hospitals. So yeah. So New Mexico has had a rapid increase in carbapenem resistant infections, including pathogens that have been carrying this VIM gene. And the New Mexico Department of Health has actually identified 183 cases of CRE between 2015 and 2017. So now in 2021, there is an increase in this, more than likely because of the abuse of antibiotics that happens. So last year, we were actually informed that the New Mexico Department of Health identified or determined 4 cases of antibiotic resistant infections with no known healthcare source. And the only commonality between all of these patients was actually where they resided, and specifically their counties of residence. So these were DeBaca, Chavis, and Eddy. And due to privacy, we weren't able to obtain the genes that corresponded with these infections, but that's what I'm looking at. We're looking at the environmental source. So usually, right, we as scientists and engineers ask, why do we care? And so for my project specifically, we care about this because we want to understand the role of the environment as a reservoir of antibiotic resistant, which could give insight in the prediction to predict and control potential outbreaks. So 6 carbapenemase genes were screened. This was VIM, OXA, NDM, IMP, KBC, and GEST. Below, sorry, it's hard, I don't have it there, but the figure to the left down in background actually shows the sample sites. These sites vary. So sample sites 1 and 2 are closer to Carlsbad. Sample site 3 is a lake right outside of Carlsbad. Sample site 4 is actually near Artesia. And then sample site 5 is near Acame. And so how I was able to do this is that I actually went over there over the summer and collected grab samples of the surface water from each of those locations, came back, filtered those samples through a manifold in 24 hours, and then I was able to identify and quantify coliforms using these special little plates that we use. And then I extracted DNA from that as well, and then looked at and verified the presence or absence of the several carbapenemase and virulence genes through PCR. Moving on to my results, the top gel, this is a picture of a gel, so this is how we're actually able to visualize if there was presence or absence of these genes. On this gel, it was shown that there was a presence of the VIM gene on sample site 4. NDM was present in sample sites 1, 3, and 5, and then JES was present in sample site 1 and 5. There was presence of OXA48 in sample sites 3 and 4. However, the limit of detection was pretty high, and we think that there was probably dilution factors that happen with this because we're getting environmental, sources. So it is a little bit harder to deal with all those external factors. And then below is the virulence genes. I know I didn't talk too much about it, but those are another important class of genes that we need to be looking out for. And I showed presence of EXHA in all of the sample sites of cross. And then moving on to the table, actually, there's not really a good general amount of coliforms throughout any of the sample sites. It did vary. However, fecal coliforms actually has shown to correlate with the amount of human activity around there. So for instance, at the water park at like sample site, I believe 1, there were more fecal coliforms there. So it does range, but it did correspond with human activity. And so just to give you, my important findings so far, VIM was present in sample site 4. Sample site 1 and 5 show the presence of both NDM1 and JES, and NDM was also present in sample site 3. Sample site 2 did not harbor any of these carbapenemase genes, and all of these sites range in the presence of gene, possibly due to water flow. So for instance, Lake Avalon, possibly had more of these genes and transfer of these genes because it was standstill, but we would have to look further into that. And in general, there seems to be a decrease in coliforms in the south direction as well, with the correlation of human activity with fecal coliforms. Yeah, and with that, I would like to open up for questions.
Great, thank you, Cassandra.
Thank
Really
you.
interesting.
Sorry, it was
time
super fast.
No, it was good. I think we have time for a couple of questions. I have one, if you don't mind. So you're seeing all of these I'm not going to say this right, carbapenem resistant bacteria is sort of out circulating.
Mm-hmm.
Is carbapenem resistance costly at all? And if so, then what are they? Is there just a bunch of antibiotics in our water that is maintaining this in populations?
So, okay. There are, especially in New Mexico, there are a lot of cattle like flow off and things like that. So I would say there are a lot of antibiotics present in and around our surface water. So we do have to be careful with this. And I'm sorry, what was the first part of that question?
Well, so that sort of answers it. I was mainly asking, where is this selection coming from to maintain this resistance? But if there's just antibiotics floating around, then yeah, yikes.
It's a big problem. It's pretty scary. So yeah.
Great, thank you. Any other questions?
I have a question, actually.
Yes,
Suraj, make it a little quick though.
Yeah, so I noticed you use 0.45 micrometer manifolds. Are these just like the average size of a bacteria or a microbe is like 1 micrometer? Are these just really small bacteria you're looking at?
So we do this to filter out anything big. So like eukaryotes, dirt, sediment, things like that usually. That's the reason. They should be able to flow through pretty good. If we want it to look like for viruses, we would have used a like 0.2. But I wasn't looking at that. I was just looking at bacteria. So
Got it. Thank you.
yeah, of course.
Cool. Thank you, Cassandra. All right, let's move on to our next presenter. And that is Catherine Persinger. She's going to be presenting on behalf of her co-authors, the characterization and host determination of 4 novel phages for potential use in phage therapy. Pretty cool. All right, Catherine, go ahead and take it away.
Great, thank you. Thank you, Joel. So yeah, like he said, I'm going to be talking about phages. So a little bit of background, like we've seen in the past 2 presentations, antibiotic resistance is quickly becoming an increased risk to human health. So we need to find some maybe alternative methods to antibiotics, and that's where phage therapy comes in. So what phage therapy is the therapeutic application of bacteriophages to antibiotic-resistant bacterial infections. And typically in a phage therapy, in phage therapy, we would use phage cocktails, which include several phages so that they can target different aspects of the bacterial defense mechanisms, as well as reduce the potential for resistance. And there's a particular bacterium called Pseudomonas aeruginosa that has been classified by the CDC as a serious threat. One, because it produces sticky biofilms, which are pretty impenetrable for antibiotics. And the biofilms also facilitate horizontal gene transfer, allowing those antibiotic-resistant genes to flow through the bacterial community. So phage therapy is being proposed as an alternative to antibiotic resistance or to antibiotics. In this project, the DeVoe laboratory isolated 4 novel phages from Socorro, New Mexico. And our project was to determine their host range and characterize them, because in order to use phages for phage therapy, we need to know what their hosts are, what they can infect and what they can't infect. So to determine their host range, we obtained a panel of antibiotic-resistant Pseudomonas aeruginosa strains and tested our phages against those to see what could infect and what couldn't infect. So how we did that was doing spot assays, which is where we basically dilute our phages down to 10 to the negative 12 plaque forming units. And we can see kind of that process in figure 2, how we would serial dilute our phages. So after serial dilution, they would be spotted onto these lawns of the panel of antibiotic resistant Pseudomonas aeruginosa and incubated, and then we would see if any plaques have formed. And plaques are just clearings of the bacterial lawn where the viruses have infected and killed their host cells. So if we look at figure 3 at the bottom there, you can kind of see on the left side, there is, I believe that's antibiotic resistant strain 54, and it's being infected by sewage phage 4. And we can see there are small clearings at the top dilutions, while on the right side of that picture, that's a different strain of Pseudomonas, and the strain is resistant to these bacteria. So our phages, called SP1, 2, 3, and 4, were attempted to be tested for all their host ranges, but unfortunately SP1 and 2 were contaminated, so we couldn't do further testing on those. And but we did get data for SP3 and 4, and it turned out they always infected the same way or the same strains, or they did not infect the same strains. So the only difference between their infectivity was the extent to which they were diluted. So for example, if you look at table 1, the antibiotic resistant isolate 54 could infect down to the -4 dilution with SP3, but SP3 could only infect that isolate down to 10 to the -4, and then SP4 could only infect down to 10 to the -2nd dilution. So in future directions, we would of course want to obtain pure stocks of those phages and re-examine their host ranges. And we'd also want to maybe adjust our PCR primers a little bit for detection of our phages, but that's really difficult because SP1, 3, and 4 are extremely genetically similar. While 2 is a little bit different, but it's very hard to distinguish them based on these primers. And another future direction may be to look at the mechanisms of infection that these phages use to determine maybe which phages would be best to use in a specific bacterial infection. Great. Yeah, and I would like to open it up for questions if you guys have any.
All right, thank you, Catherine. Any questions about phage therapy?
I've got a question. I'm not a biology major, so sorry if this is a little basic, but what makes phages so effective against multi-antibiotic resistant bacteria?
So phages are specifically, or bacteriophages are specific to their hosts. So they have very particular mechanisms that they attack the bacterial surface with. So like, like I said, biofilms of Pseudomonas aeruginosa, they're really hard to penetrate with antibiotic drugs. So phages have more mechanisms that can be used to maybe get into that. It's a little like penetrate that biofilm a little bit more, and they just have more, I guess, offensive mechanisms to attack the bacteria than antibiotics would.
Okay, thank you.
Nice questions, yeah. Any other questions for Catherine? All righty, cool. Thank you for a great presentation and why phages matter. I think that's really cool. Yeah, potentially offering a biological solution to some of these problems where biochemistry becomes a little, they adapt to it. Very cool. So we have to go back one poster because I misread my program and I skipped over another Catherine, Catherine Bosley, who's going to be presenting a poster on ancient agricultural techniques and the effect of lithic mulch on soil moisture retention. Catherine, I'm sorry for giving you a heart attack, but we're excited to hear about this, and go ahead and take it away when you're ready.
All right. I guess I should just point out if the last few ones have kind of gone over your head, this one is basically rock make ground wet. So you're in for a treat. So we have studied the effects of lithic mulch on soil, focusing on soil moisture and temperature. Now, lithic mulch, as its name would suggest, is the application of a layer of rocks to a field in order to influence the soil beneath it. So the process has been around for thousands of years. It was originally created in ancient times by early farmers, and it's been used in different cultures all around the world. Native American people, specifically the Anasazi in New Mexico, developed lithic mulch as a technology that allowed them to farm in the really harsh climate that we have here in the Southwest. What we've done is we've attempted to replicate their work by building mulch sites and then watering them by hand and monitoring the moisture and temperature, with the goal being to figure out how much of an impact the mulch has on the soil and if the technique is still useful and relevant for agriculture today.
Hello, and I'm going to talk about how it works. It's actually quite simple and ingenious at the same time. So through the layer of the lithic mulch, you add to the soil and add water. The water becomes harder to evaporate within the soil for the rocks entrap it from evaporating. So no sunlight hits it to warm it up faster. And this helps provide a greater chance for plants to survive in high heat environments, especially in drought conditions.
Yeah. As far as how we actually did this as an experiment, we have 2 locations where we have at each site 3 plots. Now, one of these locations is in fairly coarse sandy soil and the other in dusty clay-rich soil, giving us an idea of common soil conditions in the Southwest and many other arid climates. These 2 sites, other than the soil, were fundamentally the same layout. 3 large square plots, each divided into 4 smaller subplots, with each subplot being delegated to a different combination of whether we added mulch or we added water artificially, and then ones that did not have mulch and ones that did not have water artificially added. Of course, natural precipitation was monitored for all of them because that's quite precious here in the Southwest. Each subplot, each of the small subplots, had a moisture temperature probe buried in the middle of it, which was recording continuous data on 15-minute intervals for the entire duration of this experiment, which was more than a year. And then all of that data, big old spreadsheet, was crunched down into averages by day in order to generate these nice pretty graphs so that we can figure out visually what the impact is.
Okay, moving on to the data analysis. The graphs here, you can barely see them. They show a 9-day interval, which we chose to represent due to the snowfall that happened on the 24th of March. And that big precipitation event represented a spike in water content across all plots and allowed us to record the rates of loss in that water content. Very easy to observe that. So to make sense of these graphs for you, because you can barely see them, the bottom 2 represent plots with lithic mulch. The top 2 represents no lithic mulch. The graphs on the right side represent temperature, and the graphs on the left side represent moisture content. From the temperature graphs on the right, the temperature drop was more severe on the mulched plots, regardless of irrigation. You can see the lines, both lines in each graph represents whether the plot was irrigated or not irrigated. But if we look at the moisture graph on the left, the irrigated, mulched, and unmulched plots, which is the top line, are very similar in their retention rates. However, the mulched plots that weren't irrigated are having a much easier time retaining water than the non-irrigated unmulched plots. You can see the bottom line there. After the spike, the bottom line is not going down as severely as the top graph. This demonstrates the significantly reduced watering requirements granted by the lithic mulch, which shows how important this lithic mulch would be to dryland farming as a whole.
In the future, we could expand on our findings by experimenting with different rock colors and textures, such as light versus dark rocks, to see temperature differences, or smooth versus coarse rocks. The soil types can also be expanded upon, perhaps using agricultural soils rather than bare desert ground. We may also try growing plants with the mulch to see how they fare with and without mulch. On that note as well, we did find on the plots themselves that plants did in fact grow on the plots that had the rocks, which shows that this experiment does work and we should test it further.
All right, I guess we'll open it up for questions.
All right, great. Thank you, everyone. Thank you, Catherine, Antonio, David, Daniel, and Sharon. Did I get everybody? And that was a really insightful presentation. And there was a question in the chat that was, what is the recommended, or sorry, what's the benefit of lithic versus organic mulch? And David, go ahead and provide your answer because I thought it was really helpful.
Yeah, I tried to answer that in chat briefly. Basically, gravel is really freaking ubiquitous. You know, it's just a bunch of really tiny rocks, or well, not that tiny. We can detail that later because there was a question related to how thick this mulch layer should be. But organic mulch, like you'd find in like garden stores, is a lot harder to source, especially when you have like Native Americans thousands of years ago and this extremely arid climate where you can't exactly have an efficient way of developing large amounts of organic mulch, but you still get a measurable impact from just using gravel to coat the soil. Yeah. And as far as
I can add to that.
Oh, go ahead.
Another thing is it gets really windy around here and rocks don't exactly blow away. So not only is it really easy to find because it's all over, but the rocks are going to stay in place and influence other factors like runoff. They can cause water to linger in an area longer than it would normally, allowing more water to infiltrate. And on top of that, the rock is just, it's denser, it's flatter, it's more, I guess, sitting against the ground to help trap in water a bit better. So it does have its benefits, although organic mulch can be preferable in some situations.
If we have time for that other question to
Yeah,
be
go
answered.
ahead. Go ahead. What's the recommended thickness of the lithic mulch?
Yeah, so we didn't really have an opportunity to experiment with different thicknesses. The thickness that we had was roughly, I believe, about 3 to 4 centimeters thick, depending on exactly how we leveled out the gravel. But we tried to keep that pretty much constant across all of our test plots. So this was more a study of if adding gravel at all has an impact, and perhaps in the future we can experiment with varying depths of that mulch.
Yeah, we basically raked it out to a layer of maybe 1 to 2 rocks, and the rocks themselves ranged from maybe quarter size to fist size. It was pretty coarse gravel. So in the future, we could say use a more uniformly sized gravel and try and get an actual measured thickness, maybe like 1 inch, 2 inch, and see if that has a significant difference. It might also do other things like suppress weeds. So that's kind of neat.
Awesome. Thank you all. That was a really wonderful presentation. Really great information. So thank you so much. All right, let's move on to our next presentation, which is Idalise Hernandez, and she's going to tell us about incorporating titanium nitride nanoparticles into sol gel glass. Whoa, that sounds cool.
Hi,
Go ahead and
everyone.
take it away, Idalise.
Hi, can you hear me well?
We can hear you great, thank you.
Okay, so today I would like to talk about the incorporation of titanium nitride nanoparticles into a sol gel glass. So titanium nitride is an attractive photothermal material because of its unique plasmonic properties. So these particles are able to absorb light and convert it into heat energy. Encapsulating these nanoparticles in a glassy material or even like a thin film can be applied to solar thermal materials, microfluidic devices, photovoltaic cells, and much more. So titanium nitride nanoparticles tend to aggregate in really large clusters, as you can see in those TEM pictures there. So obtaining stable and monodispersed nanoparticles is important to make use of their plasmonic properties. I have been focusing on incorporating these nanoparticles in a gel monolith using the sol gel method. The sol gel process involves like several steps like hydrolysis, condensation, gelation, aging, drying, densification, and crystallization. So right now I'm focusing on optimizing the drying procedure to obtain a crack-free, stable, and uniform titanium nitride glass. I am also working on obtaining a well-dispersed thin film of titanium nitride nanoparticles onto a substrate. The use of a drying chemical control additive has proven to be helpful to me. So I'm able to stabilize and disperse the nanoparticles in DI water using sonication, supersonication, and centrifugation. I dilute the stable nanoparticles and add them to a sol gel solution and stir them well. Then this sol gel solution is transferred to a hydrophobic container and covered with foil to gel at room temperature. After the gel is formed, the gel is soaked in the drying chemical control additive, DMF, or dimethylformamide. So after this gel is soaked, it can be dried at room temperature and then sintered to obtain a titanium nitride glassy material. These first three pictures in the middle top show the sol gel formed in the aluminum dish and then soaking in DMF and then left to dry at room temperature. So after the material, so after a long time, I dried it at room temperature. I was able to obtain a glassy material that exhibited the broad absorbance spectrum of titanium nitride, which is really promising because this just tells me that I successfully encapsulated uniform nanoparticles. The bottom picture is a sol gel that was not soaked in DMF, and it created a really brittle glass that did not dry uniformly. So this is just letting me know like the DMF is really helping me out. So I did attempt to sinter a wet gel in a nitrogen environment, but I learned that the sol gel needs to be like completely dry before sintering. So I need to work on that. And I also have been using a dip coater to dip glass slides into a nanoparticle sol gel solution and obtain a thin film that shows the strong absorbance of titanium nitride. 3 coats of the sol gel solution shows like the best uniform absorbance across the glass. And in the future, I will be working on optimizing the drying and sintering processes. And to conclude, this semester I was able to successfully encapsulate stable and monodispersed nanoparticles in a sol gel. So thank you. I'll take any questions now.
Wow, thank you so much. That was so cool.
Thank you.
Any questions for Idalis?
I have a quick question, just because like I kind of know about the sol gel kind of thing. How long did it, like how long did you have to dry it for? Did you just like leave it overnight?
So when I dried my wet sol gel, I actually dried it for months. It took months to get a glassy material. And that's not unheard of. So I'm trying to optimize that procedure so I can get a glassy material in like a couple days.
Yeah.
Mm-hmm.
Okay, cool. Yeah, that's crazy. Months, like
Oh yeah.
Production wise, that would be kind of like unheard of, right? If you want to scale it up, it's like, yeah, it takes me months to dry this thing out. So, okay, cool. Thank you so much.
We have a question in the chat. What are the industry applications for the titanium nitride nanoparticle sol gel?
That's a great question. So right now I'm actually working on solar thermal materials. So I'd be using this sol gel coating to improve water evaporation. So on an industrial scale, my nanoparticles will be able to enhance water evaporation. This can be like applied to like salt production. That's just like one example. Also, the microfluidic device is really interesting. I don't know if this is like industry scale, but the microfluidic device will be able to like provide energy, just using like the sun anywhere you go. I don't know if that answers your question.
That sounds pretty cool. Do you have any ideas about how you might make this process not take months? Like, can you put it in a chamber that has like carefully controlled environmental conditions that could accelerate that? Or are there maybe reagents that you could add to the soil gel to help it dry, but would not make it brittle? Like you added the DMF and that appeared to help, right? But are there other things that you can tinker with to try and get the drying process moving along?
So I've been working on just drying them in a vacuum oven. And right now, like my best temperature to dry them out at is like 40 C. And I'm able to actually get a glass within a week that way, but I'm still working on like really getting it right, you know?
Yeah, yeah. So doable, and there are opportunities for improvement. Cool.
Yes.
Wonderful. Well, that was really, really cool. Does anybody have any final questions? One quick question we could take. All right. Thank you so much, Idalise. It was really, really great. Let's move on to our next presenter. And this is Haley Dietz, presenting on the geochemistry of, I don't know if this is an acronym or not. I'll let her say the title so that I don't ruin it. Haley, go ahead and take it away. Thank you.
Okay, so today I'll be talking about the Tahoe granite, which is uranium, thorium, and rare earth element mineralized. So that's what the acronyms stand for in the title. So the Tahoe
I was going to say
is
3, so I'm glad you said it. Thank you.
Yep. So the Tahoe granite is actually located in Socorro County, about 8 miles east of here in the Quebradas. My co-author and I decided to look at this granite because it could potentially have resources of uranium, thorium, and rare earth elements, or REE. Previous studies have shown that it's similar to the Alaskan Vulcan Mountains, as well as the Rossing deposit. Both of these areas are incredibly important resources for uranium, thorium, and rare earth elements. Uranium and thorium have a wide variety of applications, especially in nuclear power plants, and rare earth elements are very important for green technologies such as solar powers and smart cars. So the bulk of this project came from drill core, which is located on the pictures on the left. It was drilled in 1979 by the Rocky Mountain Engineering Company, who were looking for uranium. They noticed that there was this powdery green mineral that usually looks like uranophane, which contains uranium and rare earths. And so they wanted to see what was going on at the subsurface. There was about 1,800 feet of drill core, which is basically just long cylinders of rock about the width of my fist. So to characterize the uranium, thorium, and REE, I would log the drill core, and this consisted of many, many hours spent in the lab noting characteristics like any alteration, the color, mineralogy, cross-cutting relationships, and pretty much anything that made the drill core a little more interesting than just plain granite. I also prepared samples for thin section analysis and geochemistry. A lot of the results were proven by the geochemistry. It showed me exactly how much uranium, thorium, and rare earth elements there were in the granite. What's interesting about the Tahoe granite is that it is very unusual for similar granites of the same age found in New Mexico. Figure 3 actually displays this. In the 1940s, Kent Condy went through and he analyzed many of the granites found in New Mexico, and he grouped them into 4 different areas. And the Tahoe granite actually doesn't fall into any of those areas, which was very interesting. The other thing that's unusual about the Tahoe granite is it's sadly, little disappointedly depleted in rare earth elements, as shown by figure 4. The figure below that is a similar plot that compares my Tahoe granite to the Condy granite, and it shows that the Condy granite has an unusual pattern. So that was another interesting thing we found. There's also a table that compares the thorium and uranium elements to the granites that Condi also analyzed. The other thing I did was I analyzed thin sections. So I looked through a microscope, and I also tried to know any of the veining, the alteration, and the trace mineralogy. For example, I found some titanite, which is a titanium-bearing mineral. So even though this Tahoe granite is depleted in rare earth elements and uranium, it's not considered an economic source. It is, however, considered an occurrence because it is a little bit enriched in uranium and thorium and rubidium, but nowhere near economics amounts. Because of this, there's not too many future studies that can be done on this granite, but it would be interesting to characterize the paragenesis or order of events. For example, when I was looking at the drill core, I noticed multiple veining of barite and fluorite, and it would be interesting to establish what order that occurred in. In addition, something had to deplete the rare earth elements. It'd be interesting to determine what depleted those rare earth elements and if they went somewhere close by. But all in all, it was a pretty fun thing to look at. So I'll answer any questions.
Thank you, Haley. It was really great. Really cool presentation about this awesome geology. I'm going to ask a question real quick and then get out of the way for other people. You have this pretty large, an order of magnitude scale of variation in uranium across your 4 Tahoe samples, where you have the Tahoe and Tahoe 101 with lots of uranium. I mean, from my untrained eye, and then Tahoe 130 and Tahoe 113 is with very low uranium levels. Can you comment on what might be going on there, or is that typical variation that you might see in a granite sample, or what are your observations about that?
So the uranium might be so variable because uranium likes to precipitate along veins. Uranium precipitation, such as in the Grants District, is controlled a lot by groundwater movement and the material that's available for the uranium to precipitate from that groundwater. My best guess is that the samples with the higher amounts of uranium were around an area where the groundwater could flow through a little bit better, such as along a fault or some sort of fracture in the rock itself.
Oh, I was muted. Very cool. I like that idea. Cool hypothesis. All right, any other questions?
I have one that I hope shouldn't be too laborious.
Go for it,
I'm
David.
looking at all of the data here, and you mentioned that these are not economically meaningful quantities. For perspective, what would economically significant yields look like compared to this concentrations that you have?
For example, there's a very economic deposit of rare earth elements in the form of bastnasite, which is a cerium-bearing mineral. It's in California at the Mountain Pass deposit. They're mining bastnasite with anything between 30 and 60% rare earth elements, which is an incredible amount. In the Grants District, for example, with uranium, there's something like 0.13 to 0.60 weight percent uranium per short ton that they mine. So it might seem in relatively small amounts, but it's definitely a lot bigger than just a couple hundred parts per million that my Tahoe granite is reporting.
I have geology knowledge and experience, but I'm not an economic geologist. So thank you for that context.
Any other questions for Haley?
You mentioned that rare earth elements can deplete over time. What are some of the sources that cause that depletion?
Fluid evolution is the big one. For example, in the Tahoe granite, I noted a lot of fluorite mineralization. It was very pretty. It was purple and green and a little bit of blue. And fluorite can actually substitute rare earth elements into its crystal structure. So if you have a fluorite mineralization event that comes in, the rare earth elements can substitute into that fluorite, and then it might just exit out of the system by some other mechanism. And I think that might be where some of the rare earth elements went to, but there would have to be a lot more studies to show that.
Definitely, thank you.
All right, thank you, Haley. Great answers to those questions. That was really, really educational. Taught me a lot. I don't know anything about geology, and you taught me so much. So thank you. All right, let's move on to our next poster.
Dr. Sharber, it looks like you're on mute.
Sorry, thank you. Let's move on to our next poster, sorry, by Joshua Ward, Zia Dewan, Nathan Reynolds, Samuel Herrick, and McLeod Adams. Go ahead and take it away.
So, hi. So we were with the New Mexico Tech Electrical Engineering Department, and our project is, we are Team 5 in the Capstone Senior Design course, and our project is the Internet of Things power characterization for New Mexico Tech ICASA Plies town site, excuse me. So we have 5 team members on our team, and a project uses a current transformer and a voltage regulator sensor to receive data, and using a Raspberry Pi to transmit that power data through our data pipeline, which consists of an MQTT and a bridge to a cloud service called Kafka, Apache Kafka. It's a cloud web service API, and we're using that API to try to characterize our power data and predict, say, power outages and performance of that power line using predictive algorithms and different modeling, machine learning models. And so Zia is going to talk a little bit more about our hardware and whatnot. Go ahead, Zia. You're muted.
Thank you. Sorry, the host was not allowing me to unmute. So
I'm sorry. I have a terrible time with those instructions. They're not very intuitive, so I apologize, everybody.
No worries.
No problem.
You're doing great, Sarah. Don't worry.
Panically typing. So yeah, so the main thing was that ICASA came to us sort of with a very broad understanding of what their end goal was. So we worked with them to identify the means and sort of the critical components that would go into a system in order to develop these sort of predictive data characterizations of their town site. One thing that, so the first step that we took as a team was identifying and breaking down the sort of steps and the needed components that would go into each of those to form that sort of pipeline to get data from one site to another over using the Internet of Things. Another difficulty that we faced was during COVID, working from home and in the lab is difficult, especially with a system like this, where you have to test components in isolation a lot of the time. So as the rest of the team discusses things, we'll talk a lot about various components, and each of them were tested in isolation, as well as eventually combined, so that we can test them without necessarily having to be in the lab 24/7 or meeting in person where difficulties can arise. One component that I focused heavily on was the Raspberry Pi and the analog to digital converter, an ADC. Initially, we take these power characteristics, these sinusoids from the house that are specific to ICASA. We worked with Daniel Erickson, who is a ICASA specialist, and he was able to provide us information regarding the houses and town site information. And using that, we were able to identify AC/AC adapters and current transformers to then feed data, raw data, into this ADC, which takes the analog information and converts it to a digital source that we can then pass into the Raspberry Pi. The Raspberry Pi can then deal a lot more with the transporting and Internet of Things. The reason we used a Raspberry Pi over something like an Arduino, the Raspberry Pi is faster in many capabilities. It also has Bluetooth and wireless internet capabilities. So this was something specific to ICASA that they like to have in their system. I'm gonna hand it off to McLeod. He can talk a little bit more about how we simulated things as well as some of the systems.
Yeah, so as mentioned, the ADC has requirements that our sensors don't necessarily meet by themselves, as our AC/AC adapter outputs anywhere from 0 to plus or minus 9 volt AC signal, while our CT outputs 0 to 0.33 volts at 200 amps. Neither of which are really ideal for reading into an ADC or the Raspberry Pi. So we designed some scaling circuits and offset circuits for each using PSpice to simulate the circuits without having to go into the lab or buying actual resistors, op amps, and other components that were needed. So with that, we were able to design a basic non-inverting op amp circuit for our CT that just has a 10 times gain to give us that 3.3 volt input for our ADC. While for the AC/AC adapter, as we're dealing with a 9 volt AC wave, we had to first offset it and then also scale it. So that's using a drop circuit to drop the voltage down a bit. However, additionally, one extra requirement is that the ADC has a maximum current of 5 microamps, microamps. And that required us to build a current divider for each of those to drain the current so we don't fry our ADC and the Raspberry Pi as a result. So I'll be passing it off to Sam to talk about the data pipeline on the other side of the Raspberry Pi going to Kafka.
Looks like he's muted.
Looks like Sam is muted.
Okay, mic check.
Sarah,
1 2 1 2 1 2.
Yeah,
all
we
right,
can
I
hear
think
you.
we're good now.
Loud and clear.
All right, no problem. I'm going to talk a little bit about the data pipeline. We set up an Apache Mosquito broker on the Pi and adapted a Python script for publishing mock data to that broker. This 1 2 1 2. Sorry. This data was then transmitted to an Apache Kafka database using ICASA's MQTT to Kafka Bridge app. We had to configure that. It was pretty much plug and play, no big deal. Then we used the Kafka Streams API to implement some simple real-time event streaming processes, notably a running average. Nothing too fancy, but yeah, I'm going to hand it off to Nate to talk about real-world data organization, characterization.
I'm going to talk real quick about the data collection and characterization. And, in order for us to get power, we just needed to measure current as well as voltage. And in our case, we're using AC, alternating current, because we're using it with buildings, houses, which are usually wired with AC. And the current, though, is a bit easier to use the data off of it, because if you see a big spike in current, maybe you'll know someone's using the microwave or a compressor, to have that turning on. But other than that, because we couldn't get a lot of real world data to look at because of the pandemic, and we couldn't get things, I guess, tied together, we did look at example data and online, which gave us a good idea for algorithms to sort the data into actual useful information, like when things are turned on, like, when lights are on, when someone's in the house, etc. What time of day it might be because of the electricity usage, etc. But I'm going to try and finish off just saying that most of what we were looking at was just kind of like houses versus apartments, and maybe even businesses, but versus like what the electricity power usage looks like from how furnished a house might be, or different size houses. But that's just gonna be my overview for that.
So I see a question in the chat, and well, I think that kind of wraps up our presentation. We'll open it up to questions, and I do see a question: how difficult would a multi-node structure be in future designs? So in future work, the package system essentially just contained the two sensors, the current transformer sensor and a voltage AC-AC coupling sensor, as well as an ADC, which is analog to digital converter chip that takes those sensors and implements them into the Raspberry Pi. That's essentially all we need, and power to the Raspberry Pi. Everything else is handled on the software end by means of the data pipeline. That's what we're kind of calling it, which is the transition of data between the GPIO pin on the Raspberry Pi into the MQTT brokerage topic, and then through the MQTT to Kafka bridge and into the Kafka web. The other question about the Kafka server is Kafka is essentially it's a server that we are running on a separate machine at the moment. Maybe Sam can talk a little bit about the Kafka server, but essentially we are able to
put me in touch. All right, let me take this one. The Kafka server question. It's nothing too fancy. It's running locally on a laptop. The server itself, there's nothing special. It's just it's running the Kafka architecture for piping topics around. Kafka is really like the mechanism for transferring data in real time. Kafka is really event streaming software. So you take in data in real time and you act on it in real time. The server itself isn't anything fancy. Just like there's nothing special about the actual data storage itself, except for the fact that it's scalable and you can like partition it, and it's very fault tolerant. Like you're not going to lose anything when you're using it. So, but that's an architecture question. So I don't think there's anything too special about it. It's just really the architecture.
Thank you, guys. That was really great answers and a really great talk from a team. So with that, we have to move on to our next presentation, which is James Montoya, and he's going to be talking about bio-inspired drone recovery systems. So James, go ahead and take it away.
Hello, I'm James Montoya, and today I'm going to be talking about drone retrieval and how we can learn from the grabbing mechanisms of predators to be able to better retrieve drones. So a little bit of background about my research. My research into drone recovery mechanisms actually started with a drone crash, funnily enough. That drone in figure 1 crashed on top of a building that I didn't actually have access to. So I had to figure out how I can get that drone off that building so I can have my drone back. And so what I ended up doing was taking a 4-pronged hook and tying it onto a quadcopter drone and using that to successfully hook onto that crash drone and recover it off of that roof. So that got me thinking into how we can actually retrieve crashed drones. Because the normal way that's used is to have people walk over to wherever the drone crashed and manually grab it. And that works, but it's not ideal, especially when you have drones that can crash on top of tall buildings, crash into tall trees, crash into hazardous environments, like say you have firefighting drones. And so using people isn't always feasible. So I argue that using drones themselves to recover crashed drones is a better alternative that has several advantages. And part of that research is looking into bio-inspired mechanisms. Bio-inspiration is the process of looking at biological systems and seeing what we can learn through them to help improve our designs. And it's a pretty major aspect of robotics and drones. And so I was able to study predators and come up with a rough classification system for the actual mechanism these predators use to attach to their prey animals. My classification system had 8 main classifications. The first one was predators that use their hands, feet, claws, talons to grab onto their prey. The second one was predators that use their beak, bill, or mouth to grab onto their prey. The next group was predators that use their bodies. That would be like a constricting snake. Predators that use some sort of stinging mechanism. Predators that use suction. Predators that use their actual limbs to grab onto their prey. Predators that use their tongues, and predators that use glue or adhesion. And in figures 3 through 10, I have an example from every one of those classification systems shown. Figure 10 particularly shows what's called a bolas spider. That spider makes a long strand of silk and attaches a sticky glue-like ball at the bottom of it. And it attracts male moths. And when those male moths come flying by, it looks similar to like a lassoing action. And it lassos the moth, that ball sticks to that moth, and then it reels it up and kills the moth and eats it. So from studying these predators, I was able to come up with a few drone configurations that are possible to be built. Figure 11 depicts a rough configuration that would be a quadcopter with praying mantis-like arms that would hopefully be able to grab onto any crashed drones. I also came up with a configuration that could use constriction like a snake. And for future research, I think that configuration has a lot of potential because there are snakes called flying snakes that are actually able to glide from trees. And I think if a propulsion system were devised, you could potentially have a drone that could fly in a similar manner to a flying snake and use constriction to attach onto other things. Sorry, I lost my train of thought. I came up with a few configurations, and I think that these configurations have a lot of potential because the need for recovering drones is going to become very apparent in the near future as drones are being used for more and more applications in the real world. For future research, I'm studying not only how predators attach to their prey, but how predators hunt their prey in general to better understand how they track their prey, how they kill their prey, and how they eat their prey. I'm also studying anti-predator systems for future work. I'm open to any questions.
All right, thank you, James. Very cool. I'm going to ask a question right off the bat about the bola spiders. So you're right to highlight the silk. It's super cool, and the silk at the end is different from the stranded silk. It's this disaggregated silk that form. It's one of the stickiest things there. So my question for you though is, there's two parts of drone recovery: is that you have to grab it, but you also have to recover it in some usable format. So would the glue in that type of scenario ruin potentially the drone that you're trying to retrieve?
It definitely could if you were using an actual glue to retrieve the drone. For a bio-inspired drone, you might not want to use glue. Maybe instead you could use a magnet and have it dangling down on a strand. That way you could attach with a magnet and then pick up your drone. And then when you get back to wherever it needs to be, you can detach the magnet.
Cool.
The magnet also has the advantage, it can be used in trees because drones like to crash in trees, at least from my experience flying them. And if you're not careful, you can get your recovery drone stuck in a tree. Whereas like a strand with a magnet on the end, you can kind of hang over the tree and lower it in and try not to get tangled. And if you do, you can detach the strand.
Good idea. Any other questions? I think we have time for one. All righty. Thank you so much, James. Really appreciate it. Cool ideas. I think they just got to get them into action now. So now we'll move on to our next presenter. And that is Brandon Herkenhoff. And he's going to be presenting on the potential applications and integration of ground effects on amphibious drones, shearwaters, and bio-inspiration. Brendan, if you want to go ahead and take it away.
Can you hear me now?
Barely. It's quite quiet.
Let me see if I can fix my Zoom settings real quick. My apologies. Is that any better?
Much better. Thank you.
Thank you for letting me know. All right, well, good afternoon, everyone. As he mentioned, I'm going to focus on the ground effect, specifically related to bioinspiration. And for those who don't know, the ground effect is a phenomena that occurs in nature where when a wing, typically birds, gets close enough to the ground within one length of their wingspan, there's actually an increase in lift seen. And a lot of pilots describe this as a floating effect when they're landing. And basically what's happening is the tip vortices on the ends of the wing are being cut off by the ground, and it allows for an increase in lift due to the pressure gradient and things getting less disturbed. So there's a lot of different birds that use this, especially marine species like the shearwater or the albatross. And what they'll do is they'll glide right over the water's surface real close to the ground, and this allows them to reduce the energy usage in order to stay in flight so they can fly for longer distances. And this is a huge advantage in any form of aerial system, as it greatly reduces the amount of energy that's required for the system to operate. So my senior design project here at New Mexico Tech for the Mechanical Engineering Department is related to building an amphibious drone system that has an aerial and an aquatic member. And you can see a general image of that in the bottom left there, where the aerial drone carries an aquatic drone and drops it off. But as we're developing this, we're starting to look into different energy harvesting techniques that could be applied to a large range of drone systems, especially anything that's autonomous, where you can implement these energy harvesting techniques to greatly increase the efficiency of your flight and thus the mission duration through flight time. And this is incredibly critical for remote operation of these vehicles, such as space drones, for example. You know, they can't just run out of energy and then, you know, fall out of the sky when their battery runs out. And this would be one technique to greatly improve their potential operational duration and lead to things such as semi-perpetual flight. So this research is fairly early on in its development, but we've looked at a few wings and compared distances in terms of the lift based on the angle of attack and the distance from the ground, looking at the lift coefficient, the drag coefficient, things of that nature. And we want to start developing control systems for autonomous drones so that they can precisely fly close to the ground without worry of impact. And that's the general gist of this research. So I'd be happy to answer any questions anyone has regarding that.
All right, thank you, Brendan. Does anybody have any questions? If I could start out, I am a little, I'm not an engineer, obviously, so I'm a little confused about the amphibious design. Can you walk us through that, about how that works a little bit?
Absolutely. So that's another bio-inspired concept, but essentially you have an aerial drone member. I'm actually in the lab to work on that today, but the design concept that we've come up with is a fixed wing, which allows us to increase our flight efficiency. So we don't have to have a multi-rotor system constantly providing thrust in an upward direction to keep the system aloft. And basically it has vertical takeoff and landing capabilities. It can land on the water and operate in the air. And there's a magnetic attachment system, which you can kind of see in the bottom left. And you can't turn on and off normal rare earth magnets. However, you can take advantage of the path that their magnetic field takes and able to disable the magnet essentially. So the magnetic field no longer passes in an area you want. And this is the mechanism we're using to grab on to our aquatic member, which is a small submarine-based design. And it can carry this and then detach it, release it into the water, where it can go and do things like species monitoring or checking the health of coral, or looking at the hull of a boat, inspecting it, structural health monitoring of bridges, things of that nature. The aerial drone will sit on the surface of the water waiting for that to finish its mission. Once it has, it'll surface, send out a signal saying, here I am. And the aerial drone can then take off and pick up the drone using the same magnetic mechanism, which doesn't require active power. So whenever you're not using it, whenever you're not actuating it, you're not using power there. So there's more energy savings, and then return to whether it's a drone hub or a base of operations to finish its mission.
Oh, that's so cool. Awesome. Any additional questions? We have time for one or two more, I think. Okay, if there are none, then I'd just like to say thank you, Brendan, for a great presentation. Well done. That was very interesting stuff. And we'll move on to our final presentation of the day, which is Elijah Pulofsky. And Elijah will be presenting on cybersecurity topic classification using community labeled internet text sources. Elijah, go ahead and take it away whenever you're ready.
Okay, awesome. Can you hear me clearly?
We can hear you great.
Okay, awesome. So this topic is going to be a little bit different than most of the other presentations. That's surrounding cybersecurity and machine learning and computer science. So for cybersecurity threat detection applications, one approach is to analyze the massive amount of openly available text on the internet for relevant cybersecurity discussions about, say, a hacking event that occurred. However, it is very difficult to manually go through and read all of this text for finding discussions that mention these cybersecurity events that are relevant. That takes a massive amount of time and manpower. Therefore, we want automated methods to gather internet text and then classify it as being either cybersecurity related or not cybersecurity related. So an automated way to filter out text. In this research, we apply machine learning classifiers to this task, and specifically we create a Python tool called CTC, which can classify a large number of documents efficiently. We tested a total of 5 different machine learning classifiers trained on 3 distinct sources of internet text, those sources being Reddit, Stack Exchange, and a research paper repository called Archive. The figures shown on the poster are some examples of parameter optimization experiments that we performed in order to make sure that the machine learning classifiers were as accurate as possible. In particular, these plots show error rates as a function of the minimum allowed word length. So in general, we see a trend that the error rate goes down as a function of word length in the training documents, which makes sense because if you think about trying to classify a sentence that's maybe 3 words, you don't have a lot of context information. But if you have an entire paragraph of text, it could be pretty straightforward to see if that paragraph is cybersecurity related or not. So these results intuitively make sense. I'm not going to go into all the details for the machine learning methods right now. If you have specific questions or want me to dive into that, I certainly can. I guess in general, yeah, let me know if you have any questions. One of the, there were a lot of different parameters that we had to optimize in this research. And it can be a little bit arduous to try to explain all of these and maybe a little bit boring. So, but yeah, let me know if you have any specific questions.
All right, thanks, Elijah. I have a question, and then it looks like Samuel Herrick has a question as well in the chat. So he says, so are these algos just checking for keywords? Can I pipe the output into an RSS feed, etc.?
Okay, yeah, so these algorithms are not just checking for keywords, although essentially so the method that we use is just called bag of words. So what you do is you take your document and you sort of count how many times each word occurs. And so you get some numbers out of that, right, a vector that then you can pipe into a machine learning algorithm and train it and classify it. So it's not just keyword detection, and you can pipe the output into an RSS feed, although I haven't done any experiments with that, but it is scalable, which, like, for example, you can classify, thousands or tens of thousands of documents in a decently efficient manner. So you can integrate it with other methods.
Cool, thank you. And then another question in the chat from Gilberto. What is the main advantage of the machine learning algorithm versus a deterministic one?
Okay, so I guess a machine learning algorithm could be deterministic. It doesn't necessarily have to be probabilistic. That said, I guess the main advantage is with using machine learning algorithms compared to something more simple like keyword detection is it can take into account sort of associated words that aren't necessarily specifically a cybersecurity word. So for example, a name of a specific hacking tool, if you can detect that in some discussion, that's probably cybersecurity related. However, if you have the context information of essentially the frequency of the other associated words, you can get a better idea on a really large scale, like on the scale of millions or hundreds of thousands of documents, if there are consistent patterns in detecting this type of cybersecurity discussion. And in particular, if you can train on a specific forum, which we did do, so for example, on Reddit, then you can, hopefully the idea is the machine learning algorithm will be able to pick up on really specific patterns that maybe a manual sort of grader wouldn't be able to pick up on.
Cool, thank you. And David Cox says, I presume there are clarifying systems anywhere there is fax, tech, internet traffic, digital phone network already in existence. The 5 classification algorithms may well be able to cross over languages, equations, etc., right?
Okay, so the 5 classification algorithms, the way we vectorized the input text was using a fixed English dictionary. So I believe it was the top most common 25,000 English words as the vectorization. So these algorithms that are trained cannot cross over other languages or equations, that sort of thing. That said, you can definitely add other features besides those fixed English words that we used. You'd have to go in and retrain the algorithms, but you can do that. In particular, you can vectorize using other things like numbers or non-English text as well. Certainly that's possible, but we didn't do it for this research.
Thank you. And it looked like Zia had a question.
Yeah, I've got a question. You mentioned context. What methods did you use to minimize the amount of words that were required in order to get some context?
to minimize the number of words.
Yes, you mentioned that like it was like a paragraph versus only 3 words in a sentence. How did you optimize your system to be able to quickly read a paragraph and identify keywords?
Sure. So in short, well, okay. So the difference in classification timing, like the amount of time needed to classify a document, doesn't change in our tool. It doesn't change whether your input is 3 words or a paragraph. It's the same. And the reason is because the vectorization process turns each document into a fixed length vector. That said, the number of words is certainly important. In particular, those plots in the poster show that. If you have like a single word, I mean, that's not necessarily going to be cybersecurity related or not. That's not really a discussion at all. So for training the tool, we filtered out documents that were not basically sentences. 20 words is essentially the average for the number of words you need to form a sentence. So we filtered out documents that were less than that. And we did experiments such as the ones on the poster, right? So showing that you want those longer word lengths to get a better context. And the other thing is that the 3 sources that we trained on were very different in word length. So for example, Archive, which was a research paper database, had on the order of thousands of words per document, whereas Reddit was something on the order of tens of words per document, which makes sense if you go through Reddit. So what that means is that this particular tool, those models that we trained had, they have the context, quote unquote, i.e. they've seen documents of varying length. So they've seen archive documents that are really long, and they've seen Reddit posts that are really short. And so that's basically the answer to your question is more data, like more data from different sources sort of makes the models more robust to seeing new text.
Okay, thank you.
All right, we have one last question in the text from Sarah at SRS. She's asked, did you come across anything you weren't expecting or was surprising?
Yes, definitely. I'm trying to think of a really good example though. Yeah, I guess so, the plots shown in this poster are 3 out of many dozens that I've generated for this research. So in general, I would say the most interesting behavior is the difference among the different classifiers, which you can't necessarily see in these plots. I mean, you can sort of see it. Those plots do look pretty different. I would say that in particular, so we tested like neural networks and random forests and that sort of thing. These different machine learning algorithms behaved very differently when we varied different parameters. So for example, word length, right, as a parameter, it changes the behavior of the algorithms really significantly, which I don't think I was expecting, just how sort of strangely shaped some of the graphs can become for the different algorithms. So I think that was probably the most surprising for me.
Very cool. Thank you, Elijah. All right, well, I'd just like to say, and if everybody could unmute themselves and give the presenters a round of applause, because that was just a fantastic poster session. Lots of really great science, lots of really great engineering. So thank you so much.
Yeah, great job, everyone.
Don't miss our upcoming poster sessions tomorrow morning, and as well as Friday. And I will turn it over to David Cox to conclude our poster session.
Well, we're really pleased that you were able to be a part of this. Lots of good material and very interesting. And I'm just so pleased at how sharp you all are because it's great material that we got to listen to. I'd like to thank Dr. Charbonneau and Dr. Plant and all of those student presenters because you really made a great 2 hours for us to enjoy. Now in the chat box, you'll see that there are other events today, and that Civil Engineering's Department Showcase is still going on, and then the oral presentations will start at 5:30 this evening, and we have That's a format where there are just slightly longer presentations with a number of PowerPoint slides and then question time. And I think that will also be a fun and exciting type of session to listen to. So we hope you're going to consider some of those events and certainly keep your eyes on the calendar for future poster events and the 3-minute speech competition. So thanks again for being with us. Hope to see you 5:30 tonight, the oral presentations. Of course, stay safe, and we appreciate your work. Bye-bye.